Thank you, Kota, for your introduction. Good morning, everyone. My name is Rina FUJII, senior in立命館 University, and I belong to the Laboratory of Information of Biology. Currently, I conduct graduation study. Today, I would like to talk about a part of my graduation study using kind hybridis elegans. Kind hybridis elegans is one of nematode. My theme is analysis of Ogluck function in case of inducing endoplasmic reticulum stress. First of all, I explain about synthesizing of protein. It's essential for the life to survive in the process of synthesizing of protein. Polypeptide is fold conformation in cell. But the mechanism of folding of proteins sometimes make a mistake. It's wrong folding. To help wrong folding, the helping mechanism defolding misfolded protein. But if the amount of misfolded protein were many, it leads to bad effect. So for example, in humans, cancer, 2 types of diabetes, and so on. From recent study, Ogluck modification do some response to ER stress. Ogluck modification is one of glycosylation that provides with function of protein. This modification is regulated by 2 enzymes to catalyze Ogluck modification. Ogluck transferase, OGT, works and protein get function. To remove Ogluck modification, Ogluck-ase, OGA works and protein works. These 2 enzymes conserved metazoans from human to nematode. In this study, I tried to elucidate relationship between Ogluck modification and endoplasmic reticulum stress, and understand the importance of the Ogluck modification. My study using kind hybridis elegans. Kind hybridis elegans is called C. elegans for short. C. elegans is easy to observe a body, and his genome looks like humans. So this C. elegans often used for research. The sample was taken from wild type strain N2 in this study. As a schedule of this study, first I collected the sample which not induced ER stress in 3 stages of development: embryo, larval, stage 1, adult. Then I quantified the amount of gene of Ogluck modification by quantitative RT-PCR. Quantitative RT-PCR can compare amount of the gene. Next, I collected samples which induced ER stress. Then I quantified the gene of folding the protein. Focused on the sample which not induced ER stress and target of OGA1. Target gene is OGA1. Gene of OGA1 is remove the Ogluck modification. horizontal axis is stage of development. The vertical axis is amount of gene expression. From this result, stage of embryo were most the expression. It considers that normal or gluconac modification work in the stage of development. To make sure that I observed the phenotype in wild type strain N2 and OGA null mutant. From observed, compare with wild type strain, the timing of division and turn of the blastomere is confused in OGA null mutant. OGA null mutant have a lost ability to remove O-gluconac modification. So, not O-gluconac modification is confused. Next, focused on the sample which induced endoplasmic reticulum stress, and the gene of target is CRT1. Gene of CRT1 is folding protein. Compare with normal sample, induced ER stress is increased expression level. Next, focused on the gene target of OGT1. OGT1 is catalyzed the O-gluconac modification. Compare with normal samples. Sample of induced ER stress is also increased the expression level. From this experiment, I consider that O-gluconac modification required for embryonic development in addition to provide with function of protein, and O-gluconac modification mitigates the ER stress. However, this conclusion is still stage of hypothesis, so a lot of mini study is must. Next, I make sure that what protein is produced by O-gluconac modification in case of induced endoplasmic reticulum stress. And I hope that this study led to cleared human disease. That's all for my presentation. Thank you for attention.